Today, we will be discussing the recent advances of stem cell therapy in traumatic brain injury and Alzheimer's disease. We are going to run over three things: some background into stem cells and the advances in two different neurological conditions, traumatic brain injury and Alzheimer's disease. What are stem cells? Stem cells are microscopic special cells that can develop into other cell types. This includes cells found in the heart, skin, muscles, or nerves. Going down memory lane, stem cells were first introduced in the 19th century, and towards the early 20th century, the research over whether stem cells can provide a supply of new cells for the entire blood system was debated. This debate later led to multiple studies for the use of stem cells across all different classes in medicine, including neuroscience. In 1992, neural stem cells were identified in the human brain. From there, research has greatly progressed, and in 2018, scientists grew a 3D model of human brain tissue that was able to exhibit neural activity for at least 9 months. Likewise, similar research has been conducted on other neural injuries and diseases today. Now we will look at how traumatic brain injury is trying to utilize stem cell therapy for treatment. Traumatic brain injury, TBI, is a common condition that results in the destruction of normal brain function by a physical force. Depending on the severity of the force and the trauma, mild TBI can affect the brain cells temporarily, or serious TBI can result in bruising, torn tissues, bleeding, and other physical damage to the brain. These damages then can result in long-term complications or death. Two different stem cells have been found to treat neurological impairment from TBI. The stem cells include mesenchymal and neural cells. Mesenchymal stem cells are multipotent cells that are located in the bone marrow and perivascular tissues. These cells play a vital role in tissue regeneration by preventing inflammation and reforming cells in the area to replace the lost cells. In an experiment by Wang et al. 2013, it was concluded that MSCs move selectively to injured brain tissue of rats with TBI and segregate into neurons or astrocytes to repair, improving motor function of TBI. Similarly, in a research by Gao et al. 2017, they used the rat TBI model to study the treatment effect of MSCs found in the bone marrow by injecting them into mice with TBI. This was then compared to the control in the experiment, which is no TBI, to conclude that MSCs can promote neurological recovery, improve learning and memory ability. Moving on to neural stem cells. Neural stem cells, NSCs, are self-renewing stem cells that can become neurons, glial cells, and oligodendrocytes. The long-term survival of these cells injected into a TBI rat model is difficult to find, as most of the time it's rejected by the host. A study by Host et al. Produced the immunodeficient TBI rat and injected human NSCs to a TBI rat model to observe the recovery of cognitive function for 6 months. Through this research, it was observed that 9 to 25% of NSCs transplanted into the TBI model was alive for at least 5 months and separated into mature neurons and astrocytes. This study suggests a potential for transplanted human NSCs, as they may be an effective long-term treatment for neurological recovery after brain injury. In addition, the transfection of genes that encourage growth into NSCs could enhance the spreading and separating of NSCs. Despite the NSCs transplantation method, the time of transplantation is also a major component affecting the efficiency of NSCs treatment for TBI. Now we will move on to Alzheimer's disease and the possible use of stem cell research. Alzheimer's disease, AD, is a progressive neurodegenerative illness and is the most common form of dementia. This disease results in the progressive loss of structure or function of neurons, including death of neurons. So far, there is neither an effective prevention nor a cure for Alzheimer's disease. However, increasing research has evidences in recent decades that stem cell-based neuroreplacement therapies might be the new future for treatment of AD. In terms of available drug therapy, it can only improve cognitive symptoms temporarily. Also, non-drug treatments, including gene therapy, can only bring symptomatic relief and not halt the progress of AD. Since NSCs have been proven to exist in the CNS and involved in the repair of nervous tissues, the use of endogenous NSCs can be used in the injured regions, where they can multiply and integrate into the existing tract to promote neuroregeneration in the brain of AD. Also, in AD, as the disease progresses, it causes drastic biological responses after the lesion in the brain and activates microglia and astrocytes to the damaged area. These cells then produce cytokines that promote or block the inhibition of existing NSCs. Therefore, it is possible for damaged cells to be replaced from endogenous NSCs. However, studies have shown that the ability of endogenous NSCs to compensate for lost cells is restricted, in saying that most of the new migrated neurons in and near the injured area die before segregating into functional neurons. Thus, only 0.2% of the dead neurons are replaced. However, it has not been determined whether these effects depend directly on the promotion of neuronal regeneration by NSCs. Although stem cell-based research into both TBI and AD show promising results, there are many hurdles before these studies can be applied clinically. Most issues revolve around the complexity of pathophysiology and the application of stem cell therapy. Similarly, the limited sample size and control group also don't provide great results, so multi-long-term follow-ups as well as randomized trials will be beneficial. In terms of TBI and AD, the factors such as safety of stem cells, the route, time, and mechanism for injection are all important points for future studies. In AD, the role, contribution, and significance of newborn neurons in the human brain are unanswered for studying neurogenesis. In all, developing our understanding of the processes controlling the activation, migration, and differentiation of stem cells will be a critical step towards the usage of stem cells for new regenerative therapies in AD and TBI.